Hello, everyone. I am Sora Matsumoto, a PhD student in EMBL-Heidelberg. It is my pleasure to moderate today's RNA Collaborative seminar, jointly hosted by RNA Club at European Molecular Biology Laboratory, or EMBL, and RNA Initiatives at Italian Institute of Technology. And I'm moderating the first talk by Eva Kowalinski, a group leader at EMBL-Grenoble. Eva obtained her PhD in EMBL-Grenoble and Université Joseph Fourier in Grenoble, working on RNA sensing innate immune receptor RIG1. She then did a postdoc research with Elena Conti in Max Planck Institute of Biochemistry in Munich, working on the RNA degrading Suki exosome complex. And since 2017, she returned to EMBL-Grenoble as a group leader, elucidating protein complexes in RNA processing and RNA modifications. I also learned that she was recently awarded ERC Consolidated Grant with a project focusing on an interesting splicing system in parasite. And from the title, I guess she will talk about her research in that direction today. And before handing over to Eva, I want to remind everyone to use Q&A for asking your questions and not the chat. And please do not hesitate to ask questions like during the talk. And after the talk, I will read the questions aloud and Eva will answer the questions. And even if we don't have enough time to answer all the questions, she can continue to answer by texting in Q&A. And with this, please take it away, Eva.
Thank you, Sora, for this nice introduction. Thank you for being here today virtually. So I will present recent work from my lab on the sLRNA recognition by a trypanosomatid cap binding complex, which is a bit different from what you might know about cap binding complexes. So my lab is interested in RNA processing and especially RNA modification or modified RNAs. And to summarize the work of my lab, I made this scheme, and we have a keen interest in tRNA modifications, but these two upper circles represent RNA processing projects in trypanosomatids. This is a group of eukaryotic parasites. And we're using integrated structural biology. Well, someone raised a hand. I ignore this for the moment. Do you hear me? Is it okay? Yeah. So we're using integrated structural biology to tackle this question, to understand how these complexes work. Most of all, we use cryo-EM that we complement with biochemistry, cell biology, to understand the mechanisms of how these complexes work. Since I started my lab, we have already solved on cryo-EM structures of several RNA processing complexes. In the lower half, you see some of our tRNA processing work. On the upper half, these two projects represent trypanosome RNA processing, on which I will elaborate a bit further in this talk. If you have questions about the tRNA processing projects, please email me and we can I can tell you more about that. So why study trypanosomes? I mean, first reason is a medical reason. Trypanosomes and their relatives, cruci, Trypanosoma cruci, and also related Leishmania species, they cause disease and is devastating disease of economical impact. But other to the medical reason, there is a keen interest to study trypanosomatids since they branched really early on off other eukaryotes from the eukaryotic tree, and by observing the complexes and the pathways in this parasite, we can learn much about the last common ancestor. But on the other hand, since they branched really early from like fungi, metazoans, we also find metabolic pathways which are very different, and we have the opportunity to discover really new biological concepts in these parasites. And I will focus on RNA processing today, because in this group of parasites, the mature mRNAs, they look pretty much like what we know from textbooks from humans or yeast. They're capped, they're intronless, they can have modifications, they have a poly-A tail. But the way of processing to get from a primary transcript to this mature mRNA is very different. And just an example, I highlight here the mitochondrial mRNA editing in trypanosomes, where the DNA consisted, the mitochondrial DNA consists of maxi circles encoding for only cryptic pre-mRNAs that do not sensefully code for a gene, but they need to be edited to make sense, to be translatable by the mitochondrial ribosome. And for this, there exists a second pool of mitochondrial DNA, mini circles, that encode for guide RNA. And this information of the cryptic pre-mRNAs and the guide RNAs has to be united through editing to generate a coding transcript. This is done by large complexes, and my group has elucidated a module, a dimeric module of this editing machinery, which is the RESC1, RESC2 complex. It's a dimeric complex that selectively binds to 5' triphosphorylated RNAs, and only the guide RNAs, the small RNAs in the mitochondrial RNA pool, they carry a 5' triphosphate. So we could pinpoint this complex as the selection complex that selects the guide RNAs from the pool to bring them to editing, to the editing reaction, and that also stabilizes this form as a kind of cap binding complex of the guide RNA. But what I actually want to talk about today is the nuclear processing of these parasites, which is very interesting for a RNA biologist because it bears many novelties. So trypanosomes and their relatives, they express primary polycistronic mRNAs with many genes of our gene units of unrelated function on one long polycistronic transcripts. And these polycistrons have to be processed into individual coding units by splicing, and this is done through transplicing, which means that a small 5' mini exon from a separately encoded RNA is transpliced as a 5' mini exon to each unit on this polycistron, and this separates the polycistron and makes individually capped and polyanilated translatable mRNAs. You don't have to understand this whole process for today's talk, but what I want to highlight is that this spliced leader RNA, this universal 5' exon that is transferred to each gene unit, comes with a hypermethylated cap called cap4, and this cap structure is bound by a cap binding complex. So the capping and the transfer of the cap binding complex is done through this transplicing reaction. The cap binding complex has a role in this. So just to bring everyone on the same page, what do we know of the cap binding complex from metazoans? It consists of a small and a larger subunit, CBP20 and CBP80. And this is one of the first complexes that binds the nascent mRNA once it emerges from the polymerase II. So shortly after capping, this dimeric cap binding complex binds the cap, the M7G modified cap, and serves as a platform for many other factors that decide about the fate and localization of the mRNP, the newly born mRNP. This can be productive pathways, but leading finally to the export of the mRNP, or this can also be destructive pathways mediating the decay of a particle in case of a defective particle. So how does this look in trypanosomes? In trypanosomes, the small subunit of the cap binding complex was identified through sequence homology. No large subunit was identified. And then the group of Chris and Judy tagged this small subunit in the parasite and did a pull down. And with a small subunit, surprisingly, not only one subunit, but 4 other proteins pulled down. One of these was identified through homology as importin alpha. And the other 3 subunits were just named after their approximate molecular weights because sequence identity could not assign any function. Knockdown by RNAi of these subunits resulted in growth defects in parasites, as you can see here from the growth curve, the wild type in comparison to a induced knockout. So this is an essential complex. The cap binding complex is essential for trypanosomatids. And secondly, the effects could be pinpointed to transplicing defects. So this whole process of transplicing and mRNA processing through transplicing is affected, as you see here. When you do a knockdown of these subunits, you get an accumulation of the SL-RNA, so the input of the reaction, and you get a less and less product or intermediate product of the transplicing reaction, telling you that it has this cap binding complex has a really important function, vital function in the first step of transplicing, or before the first step of transplicing. One other thing to mention, is that the trypanosomes, they have a very specialized cap. They don't not only have this M7G modification, but they have a hypermethylated 5' end with the 7 methylations within the first 4 nucleotides. And it seemed obvious that this cap binding complex is special and different, might bind to this hypermethylated cap4 structure. So these were all reasons for us to set out for structural elucidation of this complex, since the cap binding complex is essential for trypanosomes. It's required for the mRNA biogenesis through transplicing. It has 3 subunits of unknown function, which is always thrilling for a structural biologist, and it's significantly different from humans. So actually, it might be a good drug target to get specific drugs targeting this complex in the parasite and not in the human. Before I present you our data, I will just acknowledge the people involved in the study. This was Harry, from my PhD student from my lab, and Hannah, a new PhD student who took over the project. Other people from my lab, and I'm very thankful to the chemists that helped us and synthesized and put a lot of work in the synthesis of this cap4 analog or cap4 nucleotide and cap4 RNA, Jacek in his lab and Ronald in his lab. And also Cameron from Bordeaux helped us with some validation experiments. So we started off with this model of the cap binding complex of 4 subunits. We left the importin alpha part, and we expressed this in insect cells. Trypanosomatids, they are eukaryotes, so we use the eukaryotic expression system. We expressed a tetrameric, a trimeric, and a dimeric complex, and you can see here on the gel how this looks like. And then we set out to solve cryo-EM structures of these different complexes. And this was done by Harold in my lab. And he obtained these 4 structures. And you see already these 3 structures, and you see already there's a problem. You might realize this dimer should be significantly smaller than the trimer, and the trimer should be smaller than the tetramer, but they all look about the same. And when assigning the sequences, you realize that one of the subunits, the CBP66, is completely missing from these structures. In cryo-EM, if you have invisible parts, you there are two possibilities. Either you have a broken particle or you have flexibility. And this is something we can assess. And actually, we've used mass photometry. This is a very sensitive biophysical method that can weigh a molecular weight at a very low concentration. And we did this for the dimer and the tetramer. And we obtained approximate the expected sizes for the dimer and also like 230 kilodalton for the tetramer as expected. So this indicates it's not a broken particle, it's rather flexibility within this particle. For assessing flexibility, we have a good way here on campus to assess this via the ESRF synchrotron to do some X-ray, small angle X-ray scattering. And we measured this, and there's quite a lot of data you can take out of such a scattering curve. First of all, you can also we could confirm the molecular weights. This data is not shown here, but you can also calculate the radius of gyration. And the radius of gyration tells you something about the compactness of a particle, which is an implicit measure of the flexibility. And the radius of gyration of the tetramer was really large, indicating that this would be a flexible particle. The SAX data also can give you a Kratky plot, and if the Kratky plot looks like a Gaussian shape, this signifies a compact particle. And if you have, like in case of the tetramer, a Kratky plot where you have one side like Gaussian and the rest a line or dissolved, this indicates flexibility. And this is exactly the case for the tetramer. So we see through the SAX data that we have a large amount of flexibility in this particle. And since we had a cryo-EM structure part of it, we visualize this in this composite model where we have a part of the complex with this small CBP20 subunit that binds the cap, which we have a cap analog in here. We see this in the cryo-EM structure. This green subunit, the larger subunit, actually from the shape and the function is very similar to the large subunit of metazoans. So the CBP110 is the functional homolog of the CBP80. And then we see part of this red subunit, CBP30, in our cryo-EM structure. And we have this flexible part attached to it with this CBP66, which is invisible in our cryo-EM structure. The CBP30 is the bridging subunit, and because there's unstructured protein, as you see from AlphaFold prediction, and we assess this a bit further. I mean, the binding region where this flexible bridge binds to this green large subunit, we see nicely in the cryo-EM structure. It's a hydrophobic helix that binds in a hydrophobic groove, which we also assessed by mutant analysis, which I'm not showing here. But to learn about the other part of the complex, the invisible part of the complex, we had to use a divide and conquer strategy where we did overexpression of all subunits in hex cells and pull down on the red subunit, on this linear, on this CBP30 subunit, just going via a frame to signify the interaction region of CBP30 with this blue CBP66 subunit. And we could pinpoint this towards the C-terminus of CBP30. Good. So much about the architecture of the proteins involved, but what is now about the cap and the RNA? So we had a cap analog in our structure, but of course we wanted to confirm this experimentally. And you can buy a resin where you have a cap analog M7-GTP bound, and then you can just pull down assays, and if your complex of interest is a cap binding complex, it will pull down in the resin. And this is what we did. And when you compare the input with the pull down, you see nicely that we can pull down all 4 subunits of the complex. But what about specificity? For the specificity experiment, we did a competition experiment. So we put other nucleotides in our pull down and expected that nucleotides that would compete with the binding would compete out with the cap binding. And we did this for M7-GTP, which we added in the reaction. And you see that we have less interaction of the complex with the resin. This is not the case if we put just GTP or ATP in the mix, which means that the complex, that the cap binding is really specific for M7G and not for other nucleotides. And this is an experiment that was also confirmed by NMR experiment with our collaborator Cameron in Bordeaux, Cameron Margaret. Good, so this was the experiment for the M7G cap, but I told you earlier that trypanosomes have this special hypermodified cap with these many methylations within the 4 nucleotides. So the chemists from the IIT lab, they synthesized for us a custom cap4, which we also put in our cryo-EM grid preparation. And to our deception, when you look at the cryo-EM density, we only found density for the M7G, so for the first nucleotide. So this is looked at the normal map level. If you ramp down the map level to a level that shows a lot of noise and what you should usually not show, we see that this nucleotide is there. So it's not hydrolyzed or so, it's there in the reaction, but it seems just to be very flexible bound, and only the M7G, the first nucleotide, the cap, is tightly bound. And if you would now look a bit more detailed in this contact region, there's also no sign that it would tightly interact. There's no positively charged residues that would coordinate tightly the RNA. So it's really flexibly linked, and there's many valence pointing towards this nucleotide. So there might be another interactor in the cell that actually needs this cap4. It's not the cap binding complex as we have expected. Good, but how does this now look in the context not only of this cap4 nucleotide, but of a longer RNA? So we also wanted to test the interaction with longer capped RNA. What we did was EMSA assays. You might be familiar with this. It's a native gel where you mix a protein complex with the RNA substrate, and you quantify the disappearance of the RNA from the input to generate some binding curves. So we did this for the dimer and for the tetramer, and since we had the observation from the structure that cap0, the cap4 is not specifically coordinated, we tested the cap4 against the cap0, and we see we get very similar binding curves here, the green and the orange curve, meaning that the trypanosomatid cap binding complex is not specific for the cap4 and can bind or requires cap0, so the M7G. But it does not bind to 5' or H sRNA, so it needs a cap to be present, what you see here from the blue curve. The surprise came when we did the same experiment with the tetrameric complex, where we have this 4th subunit, the CBP66 present. Suddenly, the complex also binds 5' or H RNA. And this indicates actually that this blue subunit, CBP66, confers additional RNA binding specificity. And these capped RNAs, they were provided by the lab of Ronald Mikura. So the CBP66 is the subunit of which we do not have a structure, and we tried, but yeah, it was difficult. So we used AlphaFold to generate a prediction, and we see this CBP66 has 3 domains that could potentially bind RNA, RM, a zinc finger, and the ATP grasp fold. And so we tested RNA binding of the CBP66, of this subunit, and we tested compared double-stranded RNA and single-stranded RNA. And from the binding curves here, you see this is again an EMSA experiment. You see that that double-stranded RNA is bound by the CBP66, while single-stranded RNA is not. We wanted to pinpoint which domain is actually important for the RNA binding. So we did point mutations of positively charged residue in all of these domains. And here you see the mutant per domain compared to the wild type curve, which is the black curve, and kind of more positive charge mutations in all domains have small effects. So it seems to be a concerted binding of RNA, double-stranded RNA of all these three domains of CBP66. This brings me already to my summary. So we generated an architectural composite model of the cap binding complex of trypanosomes, where we signified the CBP20 as the cap binding subunit, small subunit. CBP110 is a bit the homologue of CBP80 of the mammalians. CBP30 is a bridging subunit that bridges to the CBP66 subunit, which we pinpointed as a double-stranded RNA binding subunit. What comes next? Yeah, so we are working on in the eukaryotic parasite. So we want to explore now putative interaction sites for new drugs, because the complex is different from the human homologue. And then in the next step, we want to dive into what is now the role of the cap binding complex in transplicing. And this will be actually a part of this ERC project to decipher the role of the cap binding complex in transplicing and how the transplicing reaction works. And with this, I want to thank additionally to my collaborators that I have already mentioned, my whole group, my alumni from the group that have contributed to the project, our core facilities and services that we used, my collaborators, and yeah, I thank you for your attention. And I have another slide to show, maybe we can leave this on for a moment. At EMBL, we're providing a residency for women researchers affiliated to African institutions. Since this is a global seminar, this might be of interest for some of you. There are still some days to apply for a 3 months or more residency at EMBL within the infection biology. Thank you.
Thank you very much for an interesting talk, Eva. Maybe I was not completely clear about communication. So all the questions should be going to Q&A, not raising hands. And people might be a bit shy to ask questions. So maybe I will start while people are thinking about the questions. And about the double-stranded RNA, like that you show in like CBP66, was it? Mm-hmm. Yeah, how specific do you think is the recognition? Because it can bind, like it seems the complex can bind to the like stem loop without the cap structure, right? So I wonder how specific in the cell, like is there potential to like unspecifically like do the transplicing using other RNA?
Yeah. So I simplified a bit for the presentation, but the spliced leader RNA is it's a universal RNA. It's the same molecule, has a certain structure. So there is two stem loops. We have tested the sequence specificity for the CBP66 with the stem loop structure from the SL-RNA and also against crumbled, but it seemed not specific for the sequence. So maybe the CBP66 is a unit that probes several stem loops on the cellular RNA. And like this, if there's a cap, a M7G cap in close proximity, it will bind the other subunit and then create a tight binding. And these two affinities may add up. This is speculation. I don't have any data on the order of events, but this is how I could imagine.
Okay. Thank you very much. I still don't see the Q&A question here. So maybe like if you have more questions, like you can still ask questions in Q&A session and Eva can answer in the text. Okay, then with this, we can hand it over to the Italian Institute of Technology. Thank you again, Eva.
Okay, thank you very much. And hello, everyone. I'm Chiara Finocchiaro. I'm a PhD student at the Italian Institute of Technology. And today it's my pleasure to introduce the second speaker of today, which is Valeria Famà. Valeria graduated at the Politecnico di Torino in physics of complex system, and she's now a third-year PhD student in computational biology in Mattia Pellizzola's lab here in Milan at the Italian Institute of Technology. So Valeria is an expert of RNA dynamics, and during these 3 years, she developed nanodynamo, which is a method to study the subcellular dynamics of RNA metabolism. So Valeria, you can start. I remind you that you have 25 minutes, and I strongly encourage the attendees to ask questions in the Q&A panel, so not raising hand, but just write in the panel. Thank you.
Thank you, Chiara. Can you all see the screen?
Yes.
So, hello, everyone. Thanks for the nice introduction. I'm happy to be here today and to have the chance to talk about the last work we published and to estimate the dynamics of RNA metabolism. So let me start with a little background. When thinking of gene expression, one often considers RNA abundance as a proxy for transcriptional activity, but actually the very same picture in terms of abundance can be given either by transcriptional regulation, meaning that transcript A is less abundant than transcript B because its synthesis is lower, or by post-transcriptional regulation, meaning that transcript A is less abundant than transcript B because its degradation is higher. So the key idea of this work is to use the profiling of RNA dynamics in order to investigate transcriptional programs. Until recent time, the state of the art in terms of RNA dynamics profiling was given by the identification of the rate of synthesis, processing, and degradation. In this regard, let me mention INSPECT, a tool developed in the Pellizola lab that was built precisely to capture these three rates and relies on a workflow that is both mathematical modeling and wet lab experiments. One of the key experiments in this workflow is the nascent RNA profiling, which is fundamental to mirror real-time RNA polymerase activity and give us a quantitative estimation of the rate of synthesis. So in the case of INSPECT, nascent RNA has been captured by the pull down of metabolic labeled RNA, where metabolic labeling relies on the incorporation of this modified nucleoside, which is forturidine, into nascent transcript. However, the RNA life cycle is much more complicated than this picture. And until now, few work exists that tackle only specific step of the RNA life cycle. For this reason, we expanded this model and we built nanodynamo, which is an experimental and computational framework that expands our ability to quantify RNA dynamics. So according to our model, chromatin premature RNA is synthesized with the rate K1, and then it can be either co-transcriptionally processed to become chromatin mature RNA with rate K2, or it can detach and become nucleoplasmic premature RNA, which is then post-transcriptionally processed with the rate K5 to become nucleoplasmic mature RNA. This rate can also come from the detachment of chromatin mature RNA at the rate K3, and is then exported to the cytoplasm with a rate K6 to become cytoplasmic RNA. that can be either degraded with a red K7 or associated to polysome and then degraded. Of course, in formulating the model, we made some assumptions that are no nuclear degradation happens, only mature RNA is exported to the cytoplasm, and RNA which is associated with polysome but not in active translation can still be degraded. So, I said that our work relies on the combination of mathematical modeling and experimental setup. For what regards the modeling, we can write down a system of deterministic ordinary differential equation that, just to make it simple, it means that we can write how our species evolves over time in our equations. And we can see this as the forces that are contrasting. So as a balancing, for example, chromatin premature RNA is created with a force K1 and is destroyed as the combination of these two forces, K2 and K4. The same holds true for example, for chromatin mature RNA that is created with a rate K1 and then destroyed with a certain force K3. So to fit our model, we need RNA associated to the four pools, so chromatin, nucleoplasm, cytoplasm, and polysomes. We are able to get this data to fractionation of metabolic labeled RNA. So we label cell with 4SU for 20 minutes, then perform the fractionation, extracting RNA associated to chromatin, nucleoplasm, cytoplasm, and polysomes. And then we perform nanopore direct RNA sequencing, which is a method that does not require cDNA conversion. So we are able to see RNA as it is, included modified nucleoside. So we rely on a neural network called NanoED to distinguish between native and pre-existing RNA, so in silico. And also in silico, we perform the classification between mature and premature RNA, relying on the detection of electronic signal. So with this piece of data, we are able to reconstruct the expression levels for our genes. This is just an example for gene GSDP1. And with this expression level, our tool is able to infer the kinetic rates. So in this example, we can see that gene GSDP1 has a high rate of co-transcriptional processing compared to the rate of detachment of premature chromatin. Now, we use NanoDynamo to profile RNA dynamics of SUM159 triple-negative breast cancer cells. So we jointly modeled the information coming from our two replicates, and we obtained the distribution for these kinetic rates for nearly 2,000 genes. Among the purest rate, ranging from K2 to K9, we can say that the fastest one is polysomal association, and this could significantly shape gene expression. And moreover, we have the appearance of this bimodal distribution that is really peculiar because we didn't see it in simulated data. Clustering together the information coming from expression level and inferred rates, we can say that 65% of the genes have a preference for co-transcriptional splicing, while the remaining genes prefer post-transcriptional splicing. Now we want to see if we're able to capture some variations in the rates when perturbing the system. So the first perturbation we performed was the block of the impairment of the splicing by treating cells with pladenolide B, which is a drug targeting the splicing factor SF3B1. And indeed, we were able to see an increase in the premature in the intronic signal, both by inspecting genes on Genome Browser and by real-time PCR. And globally, we see that the proportion of premature reads increased by at least fourfold for 30% of the genes compared to untreated cells. So again, we can analyze our data, and following the same approach as we used for untreated data, we can cluster together the expression levels and the inferred rate. This is a differential heat map with respect to untreated, and we can see that we had a significant reduction in the rate of synthesis, this K1, an increase in premature RNA both in chromatin and in nucleoplasm. We had a reduction in co-transcriptional splicing and some switching from co to post-transcriptional splicing. Moreover, there has been an increase in the rate of export and some in also the rate of polysomal association changed a lot. So the increase in polysomal degradation might suggest the need of removing transcript that could not be properly translated, potentially through some pathway like nonsense-mediated decay. Nanopore direct RNA-seq is a useful tool also to capture poly-A tail. So we look at them and we saw that tail's length was reduced in pladenolide treated cell with respect to untreated condition. And changes in poly-A tail is positively correlated to the length of export, to the rate of export, sorry, suggesting that tail's length could impact export efficiency. We did perform also other impairment like export impairment and translation inhibition. Let me talk about translation inhibition, which we performed by treating cells with, for 1 hour with arintonin, which is an inhibitor of translation initiation. The net result of this treatment was a clear block of the translation, as we can see here from polysomal profiling. So we were not able to isolate enough RNA to sequence. For this reason, we applied nanodynamo based on a simplified model which neglects translation and association to polysome. For this reason, and for the reason that not all genes have all the species, we formulated a set of alternative models to accommodate, for example, introless genes that miss premature species, or non-coding genes, that is properly the model we used to capture the rate of translation inhibition. So again, this is a differential lead map for arintonin-treated cells. Here we can see that there has been a reduction in the rate of synthesis, and for 70% of the genes, there have been also an increase in the rate of synthesis. 40% of the genes switch from co to post-transcriptional processing. And again, these changes were accompanied by a reduction in the rate of export that caused an increase in cytoplasmic RNA. Now, I'll show that we were able to characterize the RNA dynamics in untreated cells and how they are impacted by a set of perturbation. So by looking at the differential lead map, this is the one for pladenolide treated cells, we can see that some rates varies always together in the same direction, like K2 and K3, that is co-transcriptional processing, and K4 and K5, that are the rates for post-transcriptional processing. But there are some other rates which varies always together, but in opposite direction, like K3 and K4. So we may wonder how the steps of the RNA life cycles are coupled. To answer this question, we performed a correlative analysis in which we determined the correlation between kinetic rates log twofold change with respect to untreated condition for each pair of rates. So significant correlation upon treatment with the prednisolone P are shown in this graph, where each edge displayed the correlation, and the strength and the color gives the like the direction of the coupling. So red means that the two rates are positively correlated, while blue means that two rates are anti-correlated. We can do the same for leptomycin B treated cell and for arintonin treated cell, although this last graph is only partially informative because it missed some steps of the RNA life cycle. And eventually, it came out that 92% of the coupling identified upon pradinolid B treatment were shared with, and then the same direction upon leptomycin B treatment. So here we can see a sort of consensus graph which tells us that synthesis is positively correlated with co-transcriptional processing, while it is anti-correlated with post-transcriptional processing that is anti-correlated with co-transcriptional processing. Moreover, some coupling exists also between nucleoplasm and cytoplasm, like the one linking synthesis to the rate of export and to polysomal degradation. So finally, we try to identify some regulatory factors that are potentially responsible for each of the detected couplings. To this end, we took advantage of encoded data to search for RNA binding proteins and transcription factors targeting genes that supported by a given coupling, and we performed a gene set enrichment analysis. And overall, we identified more than 100 factors, both for pradinolid B and for leptomycin B treated cell. This graph shows factors that are responsible for the coupling in pradinolid B treated cells. The vast majority of this coupling was shared. 93 factors were shared among pradinolid B and leptomycin B treated cells, and the vast majority of the factors were coming from the edge linking the synthesis to the polysomal degradation. Interestingly, this RNA binding proteins has emerged as a regulatory factor for the edge K1, K3, so linking the synthesis to the detachment of chromatin mature RNA. And in fact, it has been seen that it takes part in RNA polyadenylation. So to conclude, I show that simply using transcriptional profiling coupled with mathematical modeling, we are able to unravel the dynamics of the all RNA life cycles. That nanodynamo in particularly was able to estimate RNA dynamics in triple-negative breast cancer cells, where it suggests the importance of processing and shaping gene expression. And we were also able to shed light on extensive net of coupling between the steps of the RNA life cycles. So just to conclude, let me finish by highlighting the work in progress that for us comes pretty natural, as we as a lab are interested in the study of RNA modifications. And RNA modification, as you may already know, decorate RNA molecules and are important for RNA fate, play key role in stability. So thanks to nanopore direct RNA sequencing, we are able to see RNA modification in our data. And what we want to do is to use our tools or nanodynamo to see how the modification impacts on the RNA dynamics. With this I'm done. I want to thank my supervisor, my group, and all the people who contribute to this project, and of course you for listening. And I'm happy to take questions.
Okay, thank you for your presentation, Valeria. We have 3 questions from the audience. So the first one is from Sergio. So the question is, does your model take into account the dynamics of nuclear speckles and the cytoplasmic RNA RNP granules? So in other words, is it possible to predict the dynamics of RNA transcripts that are captured in stress granules in stress conditions?
No, we are only focusing on mRNAs, but I mean, the model can be extended by taking into account additional piece of data, like you had to have more experimental data, because with the 4 pools we have, we are, I think we reached the limit of overfitting. I mean, having more rate than this may overfit the model. So you can, if you have more experimental data coming from nuclear speckles, you can add some like rate some variation to the model to capture what you want to see.
Yeah, some more variables.
Yeah.
Okay. Then the second one is from Tristan. Concerning the correlation graph, did you try to build a causal graph like with tools MIIC Curie France, I think? I don't know. You don't? Okay.
We didn't try, no.
Okay. Okay. Maybe you can also answer to this question more deeply in the Q&A question. Okay. So the third one.
I cannot see the chat because there is on top the sharing button.
Okay. I think you can try to answer later.
Yeah.
Okay. Then the third one is from Drish. Do you only have one time point? So purify your RNAs 20 minutes after your pause? And how would you expect your rates to evolve with time for a particular transcript?
Yeah, we have one time point, and you have like here we are trying to capture the dynamics of mRNA, and the choice of time point is crucial because you don't want to have a too long time point because you cannot capture nascent species because in chromatin they evolve really fast. So if you wait too long, you're not able to capture the nascent chromatin RNAs because it will be saturated.
Okay.
So
Okay, great.
I don't know what to expect. I mean, that of course, if you wait more than 1 hour, maybe you won't be able to capture the dynamics of the species that are really near to the starting point of the model.
Yeah, perhaps you should revise your model in other terms, let's say.
Yeah.
Okay. That's it. Thank you to all the speakers and all of you for attending the seminar.